Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about immunoelectrophoresis. In the previous lecture we have already discussed about one of the electrophoretic techniques, that is agarose gel electrophoresis. Now today we will going to start with immunoelectrophoresis. So all these techniques, whether it is an agarose gel electrophoresis or whether it is an immunoelectrophoresis, all these techniques are basically used to separate two immeasurable liquid, or you can say two particles, or they basically used for the separation technique. Even we are saying that it is used for the separation technique, it is also used whenever you want to purify any particle, or if you want this application to be implemented in the uh, purification techniques. So basically the first use or the application of this technique or this electrophoretic technique is to separation, and second is for the purification. Now let us understand what this immunophoresis is. So immunophoresis refers to the precipitation in the agar under an electric field. So what is this? It is basically refers to the precipitation. So it refers to the precipitation in the agar under an electric field. So we all are aware with this term, that electrophoresis. So electrophoresis, that we have already discussed, it is a method of separation of charged molecule whenever you are applying an electric field. Okay, so basically it when the charged molecules are placed in an electric field, what they do, they migrate depending upon their net charges, size, shape and their applied current. So the velocity of movement of molecule, right, they can be further represented with one of the formula that I am mentioning here. You can just refer to this formula. So this formula is V is equals to, so here V is the velocity, okay, E is the electric field, then Q is the net coefficient, fractional coefficient. So this is the formula. I am writing it again, that V E, okay, so this is the formula. Which formula? The velocity of movement of molecule, so it is represented with this formula. Now moving to the immunoelectrophoresis. So immunoelectrophoresis, it also works like the agar or agarose gel electrophoresis that we have discussed in our previous lecture. You can refer to the agarose gel electrophoresis for better understanding. So it is used for the separation of proteins based on the charges. So this is also helps in the separation of protein, but depending upon their charges. So the mass ratio and their antigenicity, so the first protein are separated, then they are allowed to react with the antigen through diffusion or via gel, and this is known as immunoelectrophoresis. So it is very simple. Immunoelectrophoresis, so it is a process of combination of immuno and diffusion and electrophoresis. So as the name suggests, if you can see the name, so as the name suggests, it is immunoelectrophoresis. So it is a combination of immunodiffusion and electrophoresis. So an antigen mixture is first separated into components part by electrophoresis, then they are tested by double immunodiffusion. So further, these antigens, they are placed into the well, that is there, well cut in a gel without antibody. Now here you don't need to put in any antibody. And electrophoresed, a trough is then cut into the gel into which antibodies are placed. So the antibodies, now what they do, they start diffuses laterally to meet the diffusing agent. Then further what they do, the lattice formation and precipitation occur. After the formation of lattice formation and precipitation, they occur the permitting the determination of the nature of the antigen. So the term immunoelectrophoresis was first coined by Graber and Williams in 1953. Sometimes in interviews also, sometimes in any respective examination in MCQs, this questions generally arises that who first coined immunoelectrophoresis. So it was first coined by two scientists, that is Garberl and Williams, and the year was 1953. So in short, if we understand immunoelectrophoresis, it looks somewhat like this. So basically you can say it works like an agar or agarose gel electrophoresis. In this, it is used for the separation of protein based on the charge, that is mass ratio and their antigenicity. In this, what is happening? First, protein are separated. Once the protein gets separated, then they are allowed to react with the antigen through diffusion via gel. This is known as immunoelectrophoresis. Now moving to the principle of immunoelectrophoresis. So whenever we are talking about the principle of immunoelectrophoresis, then when an electric current is applied to a slided layer with gel, the antigen mixture placed in the well, what they do, they get separated into individual antigen component according to the charge and size. Following electrophoresis, they get separated. Antigens are reacted with specific antisera placed in the trough. Parallel to the electrophoretic migration and diffusion is allowed to occur. Antiserum present in the trough moves towards the antigen component, resulting in the formation of separate precipitin lines in 18 to 24 hours, each indicating reaction between individual protein with its antibodies. So this is the principle of immunoelectrophoresis. So what is the principle of immunoelectrophoresis? So basically it required an electric current, means whenever the electric current is applied to the slide layered with a gel. So what is happening, the antigen mixture is placed in the well. Then they get separated into individual antigen component according to the charge particle. So you must be aware of this principle. Now as we have discussed about what is immunoelectrophoresis, and we have discussed about the principle, let us understand that how it is happening, means what is the procedure of immunoelectrophoresis. So every techniques have their individual procedure or steps of separation, but the principles remain the same, because principle is same for every phoresis technique. Agarose gel is prepared on a glass slide put in a horizontal position. So the first procedure is you take the agarose gel, you prepare it on the glass slide and you put it in a horizontal position. Then after that, using a sample template, well are blown on the application zone carefully. Okay, after you prepare the agarose gel, you place that gel on a glass slide and you put it in a horizontal position. After doing that, you take that sample template, will are blown on an application carefully. Then you have to be very carefully while using a sample template. So now the sample template, they get diluted with 2 is to 3 ratio with protein dilution sample, that is 20 antigen solution and 20 is a diluent. So using 25 pipette and 5 ml of control, and the sample is applied across each corresponding sheet, that is known as control shield or the sample shield. Then what is happening? The gel is placed into the electrophoresis chamber with a sample on the cathodic side, that is on the negative side, right, and the electrophoresis run for 20 minutes per 100 volt. So we have already discussed the starting 2 points. So after that, what after using the sample template very carefully, the sample get diluted with a ratio of protein diluent solution, that is 2 is to 3. Then you use a pipette of 5 ml. Then you use a pipette. After the using of pipette, you take the you use it on the sample. Then the gel is placed into the electrophoresis chamber that we already have discussed in the previous lecture. Then using the then what is here, the gel is placed in the electrophoresis chamber with a sample of on the catholic side, means on the negative side, and the electrophoresis run for 20 minutes at the current of 100 volts. So for 20 minutes you run the electrophoresis for 100 volt. Then comes after electrophoresis get completed, 20 ml of the corresponding anti-serum is added. Then you add 20 anti-serum into the trough in the moist chamber, and you what you do, you incubate it for 18 to 20 hours. After incubation at 18 to 20 hours at the room temperature, in the horizontal position. So now the agarose gel is placed on a horizontal position, and you what is done, dried with the bloster sheet. Blow means you take blotter sheet and you dry those agarose gel that is present, that is placed in a horizontal position. The gel in the saline solution is soaked for 10 minutes. Then what you do, you take the gel in the saline solution and you soak it for the 10 minutes, and the drying and washing repeated twice again. So then you do drying and washing, and you have to do these two step 2 times. Okay, the gel is dried at a temperature that is less than 70 degrees centigrade. Means when you want to you want to dry the gel, you take the gel and you dry the gel at the temperature of 70 degrees centigrade, and then it may be strained with the protein staining ink solution for 3 minutes. Then you take protein solution straining, you stain in that gel with the protein staining solution for about 3 minutes. That is followed by decolorizing the gel for 5 minutes in the destaining solution bath. The gel is dried and the result are evaluated. So this is the procedure. Here 2 or 3 things you have to keep it in the mind, that at which temperature the gel is dried, that is 70 degrees centigrade, for how many minutes you have to decolorize the gel, that is for the 5 minutes. Then you have to understand this and you have to memorize this, that uh, how many this how many minutes till you have to stain this protein, staining, that is for 3 minutes. Then you have to see that what would be the incubation hours. So you have to keep the gel for 18 to 20 hours in the incubation. So all these things you have to memorize, and have should be known, that it would be helpful for the interview purpose. Then comes the gel is placed in the electrophoresis chamber. Then at for how many minutes it is placed in the electrophoresis chamber? Then for 20 minutes at what is the current? That is 100 volts. So these things should be kept in the mind while understanding the procedure of immunoelectrophoresis. Now coming to the result of immunoelectrophoresis. So after procedure is conducted, what is the result? So the presence of elliptical precipitin occurs represent antigen antibody interaction. The presence of the formulation of precipitate suggestion no reaction. Different antigens can be identified based on the intensity, shape, and the position of the precipitation lines. Now, let's talk about the result. So, the presence of elliptical precipitant occurs, they represent the antigen and antibody reaction interactions, that how antigen and antibodies they get interacted. So, the presence of the formulation of precipitate suggests no reactions. So, different antigen, that is a protein, they can be identified based on the intensity, shape, and position of the precipitation line. So that is the reason in the first slide, if you can see this, that immunoelectrophoresis refers to the precipitation in agar under an electric field, refers to the precipitation that is mentioned here, that different antigen proteins, the how they are identified on the basis of their intensity, on the basis of their shape, and on the basis of the position of the precipitation line. So this is the result of immunoelectrophoresis. Now let us understand the application of immunoelectrophoresis. So the test helps in the identification and approximate quantization of various protein present in the serum. Immunoelectrophoresis created a breakthrough in the protein identification and immunology. The immunoelectrophoresis is used in patient with suspected monoclonal and polyclonal gametopathies. The method is used to detect normal as well as abnormal proteins such as myeloma protein in human serum, used to analyze complex protein mixture containing different antigen, and at the last it has a medical in it has an application in medical diagnostic use is of value where certain proteins are suspected of being absent, example hypogammaglobulinemia, or overproduce, that example multiple myeloma. So what are the application of immunoelectrophoresis? So the first application is they that it has a great application whenever you want to identify or you want to approximate quantization of various protein that is present in the serum. Then they are also used in the patient with suspected monoclonal and polyglobal gammopathies, and it has a great application whenever you want to detect abnormal proteins, for example myeloma protein in human serum. So here also immunoelectrophoresis is used to analyze that or detect that. Then they have also used whenever you want to analyze complex protein mixture containing different antigen. They are also used in medical diagnostic use, where certain proteins are suspected of being absent or overproduced. So these are certain you can say applications of immunoelectrophoresis that you should always be known. Then let us understand that what is the advantages of immunoelectrophoresis and what are the limitation of immunoelectrophoresis. So whenever we are talking about the advantages of immunoelectrophoresis, so immunoelectrophoresis is a powerful analytical technique with high resolving power, as it combines the separation of antigen by electrophoresis with immunodeficient against antiserum. The main advantage of immunoelectrophoresis is that the number of antigen can be identified in the serum. So every techniques have certain advantages to a certain disadvantages, or you may call it as a limitation. So whenever we are talking about the advantages of immunoelectrophoresis, so it is a very powerful analytical technique, one of the most powerful technique. Whenever you want to resolve a high you want it has you can say it has a very high resolving power. Whenever you want to detect the combination of the separation of antigen by electrophoresis, so this is the main technique to be used. It also has a great advantages where you want to detect the number of antigen that is present in a serum. So these are the advantages. Now coming to the disadvantages, or you may say that the limitation of immunoelectrophoresis. So immunoelectrophoresis is a slower, less sensitive and more difficult to interpret than the immunofixation electrophoresis. So this technique is a little bit slower, and it is very less sensitive, and it is more difficult to interpret. Means if you want to detect something, so the interpretation power is a little bit difficult. And then if you compare it with the immunofixation electrophoresis, that has a good interpret spirit, then like immunoelectrophoresis, it fails to detect some small monoclonal M protein. So if you want to detect certain monoclonal antibodies like monoclonal M protein, so it is unable to detect and some of the monoclonal antibodies, for example M protein. Why? Because the most rapidly migrating immunoglobulins, so that is present is in the highest concentration, and they occur the presence of M proteins. So this is one of the limitation of monoclonal electrophoresis. Now coming to the third limitation, that is the use of immunoelectrophoresis in food analysis is limited by the availability of specific antibodies. So whenever you want to use this type of immunoelectrophoresis in the analysis of food, so it is a quite not possible to analyze the food analysis because of the because it is limited by the availability of a specific antibody. So these are certain limitation of immunoelectrophoresis. So that brings to the end of the lecture. Now let's quickly understand that what all we have covered in this lecture as so far. So in this lecture we have discussed about the definition of immunoelectrophoresis. One thing I just wanted to mention here, that you should note it down somewhere, in spite of writing the whole paragraph of immunoelectrophoresis, you just write what I am writing on the blank sheet, that if in an exam they ask you what is immunoelectrophorecusing, so you can write it here, that it is it is it is used for separation. Okay, it is a separation, or I'm just write it in, it is used for, oh, just a second, go on, I'm just writing this again. So it is used for separation of protein based on charge is to mass ratio and their antigenicity. So this is the thing. You can just note it down, or you can take a screenshot of this. This is a short definition of immunoelectrofocusing, so you may write this whenever it is asked. Okay, so this is the definition that we have covered so far. Now moving to one of the thing that I'm just mentioning here, that this immunoelectrofocusing, who was the scientist who coined the term, you must be aware of this thing. Then this is the whole diagram, that how where matlab, how it this setup look likes. Then the principle, then what is the principle of immunoelectrophoresis. Then this is the procedure we have discussed about the procedure. So you must be known about this what is the incubation period, that how much hours it will take for incubation. So you must be aware of that. You must be aware of the temperature, at which temperature the gel is dried, that is 70 degrees centigrade. You must be aware of at what for how many minutes it is decolorizing of the gel take place, that is for the 5 minutes. Okay, so you must be aware of all these things whenever you are revising this ppt. Then this is the result of immunoelectrophoresis, means what is the result, and how it is identified. So it is basically identified on the basis of intensity, shape, and position, or and precipitation line. Now we have at last we have discussed about the application of immunoelectrophoresis, means where they are used, means how they are used, and where they are used, you can say. Then at at last we have discussed about the um, advantages and limitations, means what are the advantages of using immunoelectrophorecusing, and what are the limitation of using immunoelectrophorecusing. So these all things that you should always keep in mind while you are revising this lecture. So I hope you have understood the lecture. If you have any doubt, any queries related to the lecture, and if you find any slide that you have not understood, so you can simply leave your queries, your questions in the comment section below. In the next lecture, I'll be answering the queries, as well as if I found uh, any queries for the previous lecture, So I'm just here to answer that all. So thank you for watching. You can follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. You can get this app from Google Play. Don't forget to subscribe ITLS Academy. You can follow ITLS Academy on different platform. This is our website, that is www.itlsacademy.com. Thank you for watching. Please do like, share and subscribe. Thank you, students.